We're going to talk today about the two substances that store and communicate the genetic information for human and all other living organisms. The substances are called DNA and RNA. Right now, at this very moment, DNA and RNA are hard at work in the cells that make up our bodies. In what ways are DNA and RNA at work? This problem is still cloaked in mystery and is being studied throughout the world. Today, however, we will introduce the research being conducted at RIKEN to try to solve this mystery. Both DNA and RNA are molecules called nucleic acids. DNA is composed of 4 bases called adenine, thymine, guanine, and cytosine, and these are connected in a chain-like structure. These bases are arranged in pairs, with adenine being paired with thymine and guanine being paired with cytosine. They thus form 2 chains, which are intertwined, spiraling around each other. The sequence of these 4 bases in DNA holds all the genetic information needed for life. The total of all this genetic information in a living organism is called its genome. The information carried by DNA is transcribed to an RNA molecule called messenger RNA, which is then translated into proteins. These proteins are the components that make up our bodies. The word gene, which you hear often, was originally a word that referred to elements that pass characteristics from one generation to the next. Now, it is usually used to refer to a region of DNA that contains the information to make a protein. Among the proteins made from genes are special proteins called transcription factors. Transcription factors control when and how much RNA is transcribed from a gene by binding to a region next to the gene called a promoter. This is the main flow of genomic information. Scientists used to imagine that if all the information in the genome were to be understood, all genes would also be understood, and the mystery of life would be solved. They therefore desired to read and interpret the human genome, and the Human Genome Project was launched to that end in 1990, starting in the USA. European, Japanese, and Chinese researchers participated in this project. The Japanese research institution that played the main role in human genome analysis is RIKEN. The USA took the lead in the Human Genome Project. Complementary to the Human Genome Project, at RIKEN, Dr. Akiyoshi Wada thought it was necessary to conduct research that took a more holistic view of the organism. Dr. Yoshihide Hayashizaki advocated the ambitious research project of directly studying RNA rather than genomic DNA, allowing us to see which parts of the DNA are actively being used. Even if we are able to estimate the number and types of genes that humans have, based on the Human Genome Project, we cannot know the genes that are active in each cell. Dr. Hayashizaki considered that we had to know which regions of DNA are actually transcribed to RNA in an actual organism in order to comprehensively understand the mechanisms of nature life. RIKEN decided to collect and sequence RNA from various types of mouse cells. However, a number of problems remain to be overcome. For example, RNA molecules are too fragile to handle. It was therefore necessary to copy RNA sequences to DNA and read that DNA. This DNA is called cDNA, complementary DNA. However, at that time, no technology was available for copying an entire strand of RNA to cDNA. This was one of the main reasons the Human Genome Project focused on DNA analysis. RIKEN therefore had to develop the essential technology to obtain full-length cDNA.
Well, at first there was no method to make a full-length cDNA libraries, and then we had to start everything from the beginning. I really liked to challenge and have some member in the lab say it's impossible to do.
In addition, there were difficulties with automating the procedure. If we were to prepare and analyze cDNA by hand, it would take a great deal of time. RIKEN completed its cDNA sample preparator and sequencer in the 1990s, before the completion of the Human Genome Project. Various laboratory techniques were skillfully combined with the robotic technologies at which Japan excels. As a result, the sequencing of mouse cDNA made huge progress. However, even though we knew the sequence of RNA, its function was just as unclear as ever. We knew little about the meaning of the sequences.
システムを作ってしまえばデータは出てくるんですね。でもその出てきたデータがどんな意味を持つかということを考えるというのはですね、やはり非常に高い知的な労働力で、それは我々だけではできませんでした。一流の研究者みんな集めてその意味を解読して国際的なですねデファクトスタンダード、国際標準のデータベースを作ることを目指したというのがその始まりです。でそれでファントムコンソーシアムを立ち上げることにしました。
In 2000, RIKEN launched FANTOM, Functional Annotation of Mouse cDNA, an international research consortium. Researchers from various countries met and worked together to assign functional annotations to the mouse full-length cDNA. More than 20,000 mouse cDNA strands were annotated, and the results were published 2001 in the top scientific journal Nature. This was so revolutionary that even the groundbreaking Human Genome Project used it as a reference. In FANTOM 2, launched in 2001, new technologies enabled the collecting of very minute amounts of cDNA. Thanks to this, it was possible to determine and annotate 60,800 strands of mouse cDNA, and 16,000 genes were newly identified. These increases in precision of detection have raised unexpected mysteries. The minute amount of RNA previously overlooked was found in large quantity. In this large quantity were many RNA sequences that do not code for proteins. In FANTOM 3, which was launched in 2003, these RNA sequences that carry no information for making proteins were thoroughly verified. For example, let's assume this region of DNA codes information for a protein. This other region that does not code for a protein could also be transcribed to RNA. This RNA is termed non-coding RNA. At first, the number of non-coding RNA sequences was thought to be no more than 100, because this type of RNA was considered to be exceptional. However, as research progressed, the number has been found to be at least 23,000. Non-coding RNA thus makes up approximately 53% of all RNA. This finding that there is more non-coding RNA than messenger RNA was surprising. This discovery was widely reported by newspapers and magazines, with a caption saying, "The RNA New Continent." However, some researchers expressed skepticism about the value of this research, suggesting that the findings just meant that a lot of junk had been found, and there is no biological significance to the discoveries.
ノンコーディングを出しているゲノムの領域がですね、ガラクタ DNA と言われたところから全部それが出てきた。で、いろんな調節をやっているということが分かってきたんですね。もう本当に驚きというかですね、今までみんなは何やってたんだっていうそういう立場にみんな自分たちがそういう位置にいるということをやっと認識して。
More than half of all RNA is non-coding, while messenger RNA is RNA that makes proteins transcribed from genes. What is the purpose of non-coding RNA? Why is this RNA transcribed from regions of DNA that are not genes? Further study on RNA is needed in order to get to the bottom of this mystery about life. Each one of us started out as a single fertilized egg. Our cells repeatedly divided, so that each of our bodies now consists of about 37 trillion cells of about 300 types. How do cells with the same DNA differentiate to form so many different types? In order to understand this, a comprehensive understanding of the relationships between DNA, RNA, and proteins is needed. The advent of new technology provided a big clue. This new technology is called cage technology, cap analysis of gene expression. Most RNA has a cap at its end. By capturing the capped end of RNA, the cage technology can sequence a few dozen bases of cDNA. This enables efficient investigation of which DNA regions are transcribed and to how many RNA molecules. The aim is to achieve a comprehensive understanding of the quantitative relationships of transcribed information.
The development of cage enabled us to map all of those elements, the gene switches and long-range regulatory switches. With this methodology, we can understand when and where the genes are activated.
In 2006, FANTOM 4 was launched as functional annotation of the mammalian genome. The cage technology was combined with the most up-to-date DNA sequencer, enabling a marked increase in the speed of analysis. It's now possible to investigate which RNAs are made when, and how this changes over time when cells go through changes, and take snapshots over the course of a cell's life. This is an illustration showing the 30 transcription factors represented by circles that are active when monoblasts differentiate into monocytes. A monoblast is one type of human blood stem cell which makes blood. A monocyte is one type of white blood cell derived from monoblasts. We found complex transcription factor networks change with time. This research led to FANTOM 5, which was launched in 2011. In FANTOM 5, the research was expanded to cover approximately 1,000 types of human and mouse cells. For each type of cell, RNA and the transcription control network were investigated, and a detailed catalog of the various types of cells was thus created. FANTOM 6 was launched in 2015. The objectives were a comprehensive investigation of non-coding RNA and elucidation of its functions. FANTOM has grown to be a large consortium with researchers from 20 countries and more than 100 research institutions participating. It brings the whole world together to work on such centrally important mysteries as what is life? RIKEN has now been involved in full-scale research on RNA for about 20 years. Study on non-coding RNA is now being conducted worldwide. We now understand that non-coding RNA has roles in the coordination of various aspects of biology. Non-coding RNA is definitely not just junk. On the contrary, it bears the important role of controlling the flow of genomic information rather than coding for proteins.
And we know from research that cancers and other diseases are also regulated and controlled by this non-coding RNA. So by understanding the function, we can also understand the cause of disease and potential therapeutics for disease in the future.
The non-coding RNAs work together in a network to regulate the behavior of cells, and thereby, by using the same DNA, different cell types can display the different behavior and function.
Before the launch of the Human Genome Project, scientists estimated that human beings have 70,000 to 100,000 genes. However, the project reported that there are only about 20,000 to 30,000 genes, which surprised the world. However, the outcome of the analysis at RIKEN is that the number of non-coding RNA sequences exceeds the number of messenger RNA sequences, and the number of the former is still increasing. If the general functions and actions of non-coding RNA can be elucidated in FANTOM 6, this may lead to a revision of the definition of gene. Data analyzed to date are widely open to public on the Internet. Researchers throughout the world are free to make use of these data. These data were used in the research of Dr. Shinya Yamanaka, who won the Nobel Prize in Physiology or Medicine in 2012.
ファントムがなければまず iPS の本格的な研究に至らなかったというか、山中研究室自体があのように発展しなかったと思います。ファントムっていうのは ES 細胞だけじゃなくても全身のあらゆる細胞や臓器が対象ですから、もうあらゆる研究をされている生命科学の研究をされている方が利用することができますので、縁の下の力持ちのような活動なんですね。ファントムデータがなければ私たちの研究は随分遅れてですね、きっとアメリカやイギリスで、iPS 細胞と同じ細胞が作られて名前も全然違う名前になっていたんじゃないかなと、そんなふうに考えています。今後もですね、ファントムのようなインフラストラクチャー、縁の下の力持ち的な活動が日本でもしっかり支援継続されるように願っています。
While RIKEN has proceeded with its own research, such as the FANTOM project, it is putting its DNA and RNA analysis technology and facilities to use by taking requests for analysis from around the world. The data on RNA and RIKEN's technologies are actively used in research at the cutting edge of the life sciences.
Now with genomics we can understand a little bit everything at a higher resolution, so we can have a broad view at higher resolution. With this one we can probably enter the phase of better diagnosis and better therapy.
やっと分かったと思うとですね、新たな疑問が次の世界が開けるんです。生物っていうのは多分ですね、まだまだ我々が知らないことの方がはるかに多い。
DNA, RNA, and proteins work as a complex network. It seems the mysteries of life are hidden there. What is life? RIKEN will continue to try and find answers to the questions that we humans have always held.